Hello again. Looks like this is going to be a perpetual problem. And I'm not sure there's a way around it. Go a few more minutes on the lecture and we'll call it. Okay. As I said, null alleles can also be recorded because of degraded samples or because of inhibitors cause allelic dropout. The larger the fragment, the greater the likelihood of it being dropped out. And again, we talked about ski slopes. Talked about paternity. One example of an exclusion, one example of an inclusion. Talked about the various different applications, kinship studies, sibling kinship, immigration, missing persons, identification of mass disasters. Again, the question is the cost benefits. Start talking about SNPs, SNPs. Pretty good discriminatory power. Degraded DNA may even be useful in this situation, such as the 9/11 situation where SNPs, because the fragments recovered were maybe only 50 base pairs in length and things like that, but you could still detect SNPs. More recently, SNP analysis has been used for phenotype as well as ethnicity studies. That is, you have a description of the individual without an eyewitness. There's a company that's based in Virginia, but also has research facilities in West Virginia that has pioneered some of this. They can tell the ethnicity of the individual. And in the case of some of these suicide bombers, that's about all you can work with. And by identifying the ethnicity, you can know if they came from the Philippines as opposed to Eastern Europe. And so you can focus on potential terrorists from the Philippines and ignore other parts of the world. So it's going to be a very powerful and very useful technique. Genotype frequency, frequency at each locus, frequency across all loci. I'm going to use the product rule. Here is the frequency at each locus, locus 1, 2, and so on. Multiply them together, and that's how you get those outrageous numbers. Product rule and estimates of allele frequency allow us to estimate the likelihood of two unrelated, and that's critical, unrelated individuals sharing the same genetic profile. It is not proof that the suspect is the only one who could have committed that crime. Additional evidence needs to be taken into consideration. If your prime suspect turns out to have been at a meeting at the UK when a crime was committed in New York City, then obviously, as long as you can verify that he was at that meeting, he has to be excluded. Other results, other evidence has to be taken into consideration. Oh, God. Anybody there?
Yeah, we're still here.
Yes, sir.
Well, I have no idea what happened. Okay. One of the assumptions on the STR analysis is what's referred to as the equilibrium, population equilibrium. What is going on? Some of the assumptions of this equilibrium are a large population, no natural selection, to the best of our knowledge, with the exception of some of the newer STR loci. There is no chance of natural selection. There's simply repetitive DNA. No mutations. Mutations are relatively rare. No movement, no migration into or out of the country. And random mating, which I don't actually believe humans randomly mate, except after like 6 beers on a Friday night. In terms of actual child production, I don't think it's a random event. One of the things that needs to be taken into consideration is the racial the ethnics of the subgroups and whether or not they have the same allele frequency that the larger population does. Here's an example, again, using the original 13, of a particular analysis. Shows you the frequency of the various different alleles, how frequently they occur. When you multiply these all together, you can see that the likelihood of an unrelated individual having the same STR profile is 1 in 594 trillion. Most labs would not present that to a jury because they're just overwhelmed with those big numbers, which I don't understand, but anyway, apparently that's true. Estimate of the rarity of a DNA profile. Individual, simple STR analysis, maybe not complete. You would estimate the likelihood of an individual in the population, unrelated individual in the population of having the same profile to be 1 in 130 million. Let's see how it changes when you're looking at a sibling. So it goes from 1 in 130 million to 1 in 128. Yeah, the top number is based on unrelated individuals. The lower one is based on brothers. So you can see why it's critical to include the term unrelated individuals. CODIS, Combined DNA Index System. It varies from state to state. It's one of the things about our system is law enforcement and who gets their DNA put into databases is variable, usually by state, but they're also local as well as national databases. Local tends to be large cities. The names are not included when they share data with other jurisdictions. The locals can, if they get a hit at another jurisdiction, they can contact the locals and the locals can provide the name of the individual. You have convicted offender database. You have forensic evidence database. You have, say, a rape and there's no suspect, but you can take DNA from the semen sample and put it into a database in the hopes that the same individual will commit a crime in the future for which his DNA is taken, profiled, and then you can get a hit. Missing persons database, both mitochondrial as well as STR. US, Canada, most of the world shares these databases. One interesting study, we're going to go through this and then we'll call it quits. Over a 6-year period, 14 women were raped and 1 murdered. DNA from semen, Y chromosome indicated 1 man, a single man, or at least relatives. Female DNA made nuclear analysis at that time very difficult. The serial rapist profile was deduced. Checked males in the region using interviews as well as the Y chromosome. This was in Poland. 421 suspects' Ys were checked. 1 matched. 1 of 10 autosomal alleles differed, suggested maybe the rapist was a close relative. And sure enough, DNA from the brother of the original suspect matched semen at 19 loci as well as the Y chromosome. The odds in this case of a random match was 1 in 600 trillion. Now, one more story. I said that would be the last, but let's do this one too. A rape case. Authorities were pretty certain who the perpetrator was. DNA analysis of blood didn't match the semen, though. I can't spell didn't. CODIS search. Blood DNA matched a non-twin brother. Problem was, he was already incarcerated at the time of the crime. So what happened? Any speculation? Well, it turned out that the perpetrator had leukemia when he was young. And his brother had donated bone marrow to the perpetrator. And so the brother cells were in the bone marrow, which is where blood cells are derived. White blood cells. You can't use red blood cells. They don't have a nucleus. And so they went back and they did buccal swabs, and sure enough, there was a perfect match. So this was a true case in Canada. Next time, we may talk very briefly about Thomas Jefferson and about over-extrapolating data to give a story. I will tell you up front, I think Thomas Jefferson probably was the father of several of his slaves, Sally Hemings' children. There's lots of evidence, but simply based on the Y DNA analysis, it would not hold up in a court of law. Okay, guys, I will be sending you your exams throughout the day. I hope by Monday, everyone will have their graded exam back. And as I said, if you have any questions, feel free to send me an email. Thank you all for your patience. I'm afraid this is something we're going to be encountering throughout the rest of the semester. Stay safe, practice social distancing, minimize going out. Remember, you're not just potentially endangering yourself, but relatives and other people you interact with. Especially if you're talking about grandparents, they are highly susceptible and the outcome is usually not very good. So take care, everybody. Again, thank you for your patience, and I will be talking to you on Tuesday. Bye.